Our first speaker is Katharina Simon from the University of Oxford, who actually just starts also to establish a lab in Berlin at the Max Delbrück Center. And she will talk about the role of autophagy in the immune system. Welcome.
Hello, everyone. Thank you for inviting me to this fabulous meeting in this fabulous city, in this fabulous hall and hotel. So I don't know, it's amazing. And I'm going to tell you about autophagy, and the session will be more talks on autophagy, so a very brief introduction. Autophagy is a bulk degradation mechanism of the cell whereby autophagosomes degrade bulk material and deliver it to the lysosome, which you can see in orange here. It's just a bit more in detail. The autophagy pathway looks like that, and I'm going to mention a couple of molecules which I will introduce now. So when we knock out autophagy, we knock usually out ATG7 or ATG16L1 or ATG5. It's all really the same as long as you knock it out properly. And then two other mentions here is LC3, which is a small molecule of 16 to 18 kilodalton expressed on the autophagosomal membrane when autophagy is induced. And in order to measure autophagy, we use LC3, but what you should always do is also use baphyllomycin A, which is a lysosomal inhibitor, which stops the fusion between the autophagosome and the lysosome, which means that the LC3 you're going to measure is going to be no longer degraded. So the 2 hours you have buff A on there, that's what you measure, how much LC3 accumulates in those 2 hours. Otherwise, you can't really measure autophagy. As a reviewer, we'd say this every single time when people submit papers on autophagy. Don't forget baphyllomycin A. Okay, so my contribution to the field is that about 2007-8, I decided to look at autophagy very early on. It was really discovered only in yeast. The first knockout mice were made. And I decided to look at that in the immune system, thinking that it would be a cell death pathway, but obviously turns out it's a cell survival pathway. And over the years, I have looked at many cell types, being an immunologist, in the hematopoietic system, and I'm just going to summarize this in one slide since we don't have much time, but this is our contribution to the field. We found that autophagy is really important in differentiation of immune cells, and that is not only true for immune cells, but also for most other cells in the body. That it's a requirement to have autophagy probably for a changing metabolism, but also to have enough ATP. The second major function of autophagy is the maintenance of long-lived cells. And some examples here are the maintenance of the stem cell, which lives with us for all our lives probably, memory CD8 and CD4 T cells, but also memory B cells. They are induced by vaccination or a pathogen, and then we retain memory cells to remember what we've seen before to have a quicker immune response. And the maintenance of these cells, and that I will touch upon in my talk, is maintained by autophagy, a constant clearing out of material that's no longer needed in these cells that divide very rarely, and therefore also don't dilute their debris to their daughter cells. We've also found a cell extrinsic role of autophagy. So autophagy can secrete, also called secretory autophagy, which has come up recently, and that can signal to the immune system. I won't go into that. And then the last one, which you'll be interested in, is that it prevents aging. We first discovered that in macrophages, and also in lymphocytes, but since then really looked at most immune cells, and I will tell you more about that. So the aging immune system, for those who are not immunologists here, which is probably the majority, what changes with age is that there is an increased number of myeloid cells, the so-called myeloid bias, but a reduced function. There is a reduced function of lymphocytes, T and B lymphocytes, with age. And then, meanwhile, we have chronic inflammation, which of course is mostly also probably achieved with the help of the immune system or controlled by the immune system, and autophagy levels fall. So we were always interested to find out whether the falling autophagy levels, which are not perhaps such a straight line here, it might also be a curve like this more, this is still to be looked at, whether the falling autophagy levels cause the inflammation or any of these other things that happen in the immune system. So that leaves the organism susceptible to infections, malignancy, and poor responses to vaccine at older age, and we know quite a lot about that. Not only that, the immune system also controls, we know, our organismal aging in general. So in this very seminal paper by Maria Mittelbrunn, she showed that if you have dysfunctional mitochondria and T cells, which is a kind of an aged phenotype, this leads to premature aging of the entire body. And that's probably via inflammation, via TNF-alpha. If she reduces TNF-alpha in those mice, she can see these multimorbidity, the age multimorbidity disappear. So that's one study. Another, and it's probably not the only studies, it's just two I picked out, is that if you knock out the DNA repair gene ERC1 in all hematopoietic cells, that also drives aging across the organisms with the known multimorbidities, and rapamycin can reverse the markers of senescence. So these papers really show that maintaining a youthful immune system is going to be very important for the rest of the body. And therefore, we want to find out more about how the immune system ages. So if we knock out AGG5 or 7, we find generally an aged hematopoietic system. If we knock it out in stem cells, we have reduced stemness. We get anemia. Also, if we just knock it out in red blood cells, we get anemia because mitochondria are degraded at the end of the maturation of the red blood cell by mitophagy. We have the accumulation of immature myeloid cells, excess inflammatory cytokines, and we also have lymphopenia. This is typically for an aged immune system, so all the hallmarks are reproduced. Not only that generally, but also at the cellular level, there's excess damaged mitochondria, excess lipid droplets, excess ROS. So can we show this in another way? So basically, I just take you through some more data now. In mice, when we look at autophagy levels, autophagic flux in cells, in the progenitors of the hematopoietic system, there's not so much difference between young and old, but the mature cells, T and B cells, only have half the levels of autophagy, and then also in macrophages, either stimulated or unstimulated, have quite lower levels of autophagy. So this is pretty well established, I would say. And if we just look at one cell type now, so we look at vaccine responses. The best readout for vaccine responses is antigen-specific T cells, CD8 T cells. That really tells you whether the vaccine has worked or not, as well as antibodies. So just starting with the CD8 T cells, if we look at a mouse that has got no autophagy in the T cells, so the T cell AGG7 knockout, and give it 2 shots of flu vaccination, you see that the effector response, so that will react quite quickly to the virus, is pretty good in both genotypes. But in the knockout, you have almost no memory response left. And in old, this looks quite similar, maybe not quite as stark, but there's also a severe loss of the memory response. So in this paper we published, we called it formation of memory responses, but actually, I show you later, it's probably the maintenance of these cells. So the question was, are these two related? I mean, they look similar, but is the loss of autophagy in these cells leading to this loss of memory? So the way we could do it is reintroduce AG7, which people have actually done more recently. But what we did is we just thought, could we reintroduce pharmacologically autophagy in these old mice and recover the memory responses? So at the time, there weren't many autophagy inducers known. This was 2013 or something. And only a very few had been tried in mice. And one of them is Spermidine, not being talked about yet at this conference. Maybe a good sign, maybe a bad sign, I don't know. But I'm definitely going to introduce that to you. So it's a polyamine, and it's an endogenous metabolite. Actually, we briefly heard from Tom Rando just the opposite this morning. So it's a polyamine, and it's made from arginine through the polyamine synthesis pathway, but it can also be taken up by food, and the microbiota provided too. It's known now. And this has been shown to promote longevity and autophagy by Frank Madeo's group, or had been shown at the time. And so we gave it to mice, and this is what it looks like. This is the blue and blue and black line I showed you earlier. These are the old mice. If we give old mice spermidine, it's a huge increase in memory responses, well above the young. And if we give it to mice with no ATG7, we don't have that effect. It doesn't necessarily mean it's autophagy, but when autophagy is not there, spermidine doesn't work. So this, we gave spermidine 5 millimolar for 6 weeks to very old mice here during this vaccination. And just looking at B cells, so this is, we use another model antigen, don't worry, it's for a lot of immunology, but another readout for vaccination and how well we respond to pathogens is to look at antibody responses. These are made by plasma B cells, and IgG1 titers were measured here. In the old, again, it's actually pretty poor, both effector and memory responses in the old, and this is why vaccination often does not work in elderly people, especially to new antigens that they've never seen before. And the same is true for a knockout for ATG7 only in B cells. So here too, we gave spermidine, and we could improve IgG1 responses quite well, not quite to the level of the young, but it was definitely significantly improved. So I left this quite a long time. I was in the UK, where you write a lot of grants, never had any time to really follow this up. So when I moved to Germany, I could write far fewer grants. That was very helpful. And so we were also asked by, we patented this, and they said, can you please show us it works in human? Can you please show us the pathway downstream? So we worked a bit further on this. Firstly, I wanted to show you that it wasn't actually the formation of the T cells. So a postdoc in the lab, Mariana Bosa, she took T cells that were primed with an antigen, but had an inducible knockout, weren't knocked out yet, but could be knocked out, transferred them into host mice, and either gave them tamoxifen to knock out or not knock out the AGG16L1 to show that the formation, so they were formed, but is it the maintenance or the formation? And it's definitely also the maintenance, and perhaps also formation, but it's definitely the maintenance. So if you knock it out, you can see that both genotypes have got an equally survive before they're knocked out for AGG16L1. We have a tomato red for the knockout, so we know they express this tomato red, which means AGG16L1 is deleted in these cells. But what we find is if we delete them with tamoxifen, we lose all the cells. So it is a maintenance problem of these long-lived cells, of these memory cells. Okay, so that was just a small bracket in brackets. Remember that, important for maintaining long-lived cells. So basically, as time went on and we had PhD students working on this, Hanlin found that actually a major pathway that was important in the spermidine, downstream of spermidine to induce autophagy. There were lots of other ones that had been described, but this is one that we picked up on, which was mostly shown in bacteria at the time, and that spermidine gives a moiety to a translation factor called EIF5A, and this EIF5A translates proteins that are difficult to translate. It's a translation factor that helps all protein to be translated. It sits on the ribosome. Only when it's got the moiety of spermidine, it's active. And when it's active, in particular proteins that are affected when you don't have active EIF5A, is polyproline stretches. Prolines are very difficult to make the links, peptide bonds, and so EIF5A really helps those bonds to be made. And so we then looked for PPP stretches and did proteomics and ribosome profiling, and we found it was a molecule that TFEB, I mean there were many more, but that's the one we picked out, as you do with omics. And TFEB was particularly interesting. It's actually a transcription factor with proline stretches, and it transcribes autophagy genes. So it kind of makes sense that something like that could be controlled by spermidine and turn the entire autophagosomal pathway on. Other people have shown that mitochondrial proteins, actually my PhD student that went on to work on this in Erica Pierce's lab, and then another group showed that ATG3 was controlled by this EIF5A. So spermidine has a very distinct pathway that it hits, and this turns on autophagy very specifically. So basically, We then went on to look if in human there was a problem. So TFAB is actually strongly reduced in older humans, and so is hypocyaneated ER5A, largely in all the ones we looked at, not so much ER5A itself. So we know this pathway is downregulated in aging, and so are many other pathways. So the main thing is, can we turn it on again via these pathways? Can we turn on autophagy again? So we also found that Spermidine restores TFAB and hypocyaneated ER5A and autophagy in human CD8 T cells, which I'm not going to show you due to time. And most importantly, I think for this talk, and also maybe in discussion with Tom's data, we found that Spermidine is actually reduced with age in PBMCs and in plasma. Other people had shown that. Also, hypocyaneated ER5A is reduced. These are small numbers of people because it's complicated assays. And also TFAB in human healthy PBMCs. So could we give Spermidine back to these people and restore this pathway, and so therefore autophagy, and therefore the memory response? So as I said, I stopped writing grants in the UK, so I decided to go and do a clinical trial with this. So this is my disclaimer. I'm consulting for these two companies that sell Spermidine. And we use this left one for the clinical trial. This is a clinical trial which was started in the pandemic. So we had to use COVID vaccines because that's all they were going to give us ethics for. And so we gave Spermidine when after the COVID vaccine had been given. And because I told you that it was maintenance of memory, not formation, we just wanted to see over 4 or 8 weeks after the vaccine, could we maintain the memory responses, which is usually waning, and therefore we give boosters, mostly to old people. But that was the idea. Could we maintain this antibody and T-cell responses in these people if we give Spermidine? So we gave Spermidine at 6 milligrams per day, which is much less than to the mice, but which was the dose that was tested in humans to be safe. We found it to be safe. I won't take you through all of that. I straight to the data, which is hot off the press, not in press yet. So we found that antibodies to spike protein in serum are increased in the Spermidine-treated group. So if we look after 2 weeks or 13 weeks, we see an increase. So we kind of maintain that those levels of antibodies. We think it works on the cells, on the plasma cells that make the antibodies, not on the antibodies themselves. And what we also find is that there are responders. Some respond not at all, and some respond. And we're trying to find out why some people respond to Spermidine, why some aren't. And so we're doing single-cell RNA-seq for this and trying to understand what is specific about these people. Also, we looked at neutralizing antibodies. So this is gold standards. You've got to make sure these antibodies actually do something. So we look at in percentage ACE2 inhibition and looked at different strains and found that these vaccines that have been given, mostly AstraZeneca probably because it was the first rollout in the UK for all the elderly people, that they are significantly, in all of what I've encircled here is significant. There's a significant increase in neutralizing antibodies at 2 weeks. Unfortunately, it doesn't last that long, so we don't quite know why. At 37 weeks, we've long stopped, so the last time point is 13 weeks. So this is also some data using the Aurora flow cytometry that was yesterday introduced by Eric Verden. And so here we look at 40 markers on all of these PBMCs from all these people, and we can look at CD8, we can look at surface markers, but also what do these CD8 cells produce. So, for example, the population here is the CD8 population, and within that population, we look at interferon gamma production. And then the placebo, you can see that the initially quite good population is waning, the yellow here, whereas in Spermidine it's maintained. And that's also true for perforin, which is one of the granzyme and perforin are the two killing molecules for CD8 killer cells. So they're the effector molecules. And again, here in the Spermidine group, in the responders, this is maintained. We also measured autophagy over the entire 40 people that we treated. And this is a very nice assay that we set up over the years because we always wanted a good autophagy assay that's high throughput. So we use flow cytometry. We make small holes in the membranes of the cell with a detergent. and mild detergent. The free LC31 is washed out. The bound LC32, the active one, is still hooked onto these membranes and therefore can't be washed out. And then we do intracellular staining for LC32. And then it gives you a little shift like that, and with buffet, this is the shift you get. It's often not so big, but it's a very reliable assay, which we've set up so that it would be reliable and reproducible. And so basically we find that autophagy is increased in both CD4 and naive T cells and CD8 naive T cells. This was actually the thing that made me the happiest, that you can actually see that autophagy is increased in these people. In some patients, there's also a decrease in DNA damage by gamma H2AX, by flow cytometry, and a decrease in P16, P21. So we don't quite know why that happens, but we wanted to look at some senescence markers, and so that's what we found. Again, these are the responders where this did something. Okay, I won't show you that. We also looked at TFAB levels, for which we have an antibody, again for intracellular staining, and found not very much at all in the CD8 T cells, but found some changes in the NK cells, and no changes in B cells, which was disappointing, but it's possible that in human it's slightly different, or we didn't look at the right time point. So this is really what this shows, and then we've just got some new funding to do a study which was a flu study, which we initially wanted to do really, but we couldn't do it during the pandemic. And now we're going to give Spermidine while we're vaccinating. So the people, the elderly that are over 65 will receive the seasonal flu vaccine, then we'll be treated with, we'll take Spermidine for 13 weeks, and then we bleed them again, and we do safety again, and all these assays again, and also look if this flu vaccine worked. So I just wanted to conclude here. Autophagy prevents hematopoietic aging, declines in mice and humans with age. Spermidine signals via ER5A and TFAB, and autophagy in lymphocytes, and Spermidine improves vaccine responses in mice and humans. And just to thank everybody in my lab, here's my small lab still in Oxford. Mariana did the maintenance assay, mouse experiment. This is the vaccine team, Garda and Lucy, who is very instrumental. Without them, I wouldn't be able to do this. And this is my group in Germany, and they put me in the cage up here. I don't know what that means. But anyway, so I'm funded by the Wellcome Trust, UK Spine, Kenneth Rainin, and the Helmholtz Foundation. Thank you very much.
Yeah, thank you very much for a very interesting talk. We have time for one question, maybe.
Please, I don't know if I missed it. Did you measure the Spermidine level in your study? And probably you know why I'm asking, because a couple of months ago, a study was published in which they gave 50 milligrams of Spermidine, and there was no increase in plasma level except spermin. So can you comment on this?
Yeah, that's a very good question. We did study, we did measure it. Actually, the Graz group helped us measuring it, and then it turned out that we hadn't washed off the FCS, and FCS is full of Spermidine, so we have to start again. And I don't know why it's so difficult to detect that. Even at 15 milligrams, you can't see it. So people say it's perhaps metabolized already. That, I don't know, it's difficult to measure it in cells. Plasma is maybe not the right place to measure it. So these are my answers. I'm also a bit puzzled by it, but I'm determined to find out whether we can detect it in our study.
Okay, thank you very much. I guess there are